want to do a very short bit about macrophytes and fungi. So you'll get all of your photosynthetic needs taken care of today.
He didn't answer me on his PowerPoints.
Oh, okay. You can bug him and laugh about that.
I sent him a text to make him feel better. I told him, say, hey, you need to upload your PowerPoints. He's like, okay.
Alright, isn't this pretty? That's from Pacer Pond. It's called Microsterias. It's adorable. Alright. So, some fun terminology to talk about because there are a lot of different places where you would go to collect algae. And I don't mean like a lake or a stream, those being different, but like within a lake or within a stream, there are different microhabitats that you could collect from. Remember this, Drew, from Inca Methods? Maybe? Maybe. So, we have the plankton. And where would you go to find plankton? Or what type of habitat? Let's say we were at Pacer Pond, what type of habitat within Pacer Pond, where would you go to get plankton?
The chum bucket?
The what?
The chum bug is from SpongeBob.
Oh, I don't do SpongeBob. I'm greatly annoyed by SpongeBob. What?
Open water.
Open water, yeah. So plankton's the stuff that's floating around, or some of it might be swimming around in the open water. Benthos, remember what benthos is?
The bottom.
Stuff on the bottom, yep, or stuff attached to other things, which are usually on the bottom. And have we used this term periphyton yet?
Don't think so.
Okay, so periphyton is basically the algae that's in the benthos, so algae that's attached to something. And then we have specific names for if it's attached to a rock or another plant or sand or sediment or an animal. So it's always fun to learn these terms. So epilithon, any guesses where epilithon is from? Lith is the
Oh, it's
key.
the upper.
Lith, not the epi means like upon. Like we talked about the epilimnion, that was the layer that was on top of all the other layers. So epi means upon, but lith is the key. Rocks. Yep. So epilithon is periphyton growing on rocks. And we would get out our toothbrushes to scrape a hard surface to collect algae, literally toothbrushes. It's fun, it's good stuff. We have epipelon, which is from sediments. And you might find different species in the epilithon than you do in the epipelon, you just never know. Epiphyton, any guesses where you would sample to get that?
Upon. What would fight?
Fight. What would a fight be?
Live rods.
Fight is plant. So epiphyton is algae that grows on either other algae or on submerged plants. Now we have pictures of all of these. So this top picture, it's kind of hard to tell, but can you all see if I turn the light up? Slightly close. There's some slimy stuff growing on this rock, that's epilithon. This is a sandy beach, and that color is some sandy algae. We'll talk about that name too. So this is supposed to be some plants, and then green fluff, that's epiphytic algae growing on there. This picture, we've got this green filament, and the little brown things are diatoms living on the green filament, so that's algae living on other algae. What else do we have? We have epi salmon. There is an extra P in there, which is silent. It's not a misspelling. And epi salmon is the stuff growing on sand. Epizoon, where do you find Epizoon?
Animals.
Animals, yeah. And as an example, there's we've got a lecture coming up about these little critters, but this is a cladoceran, which is a little tiny crustacean, and all these little green dots are algae living on its little carapace, its little shell. And there's one species of green alga that's only found on the backs of turtles. Turtle shells. We have another term, metaphyton, and that is algae that's not attached to anything, but it's usually associated with shallower areas where there are surfaces around. So it's also known as frog spit. It just looks like frothy green stuff. We talked about frog spit. So that's kind of floating and near the edge, but it may be associated with like some plants that might be growing there, but it's not directly attached to anything. And then we have a few algae that are endosymbiotic and live inside of generally a protist of some kind. So this ciliate, I think, it has a bunch of little cilia, has these green algae living in it, and then that algae photosynthesize and provide sugar for the cell that they live in, and the cell provides nutrients and keeps swimming toward the light so they have a source of energy.
So they have like a symbiotic relationship.
Yeah.
Hence the name symbiotic.
Well, symbiotic doesn't have to mean everybody wins.
Yeah.
That's a mutualism, because parasites and their hosts are also considered a symbiotic relationship. I didn't know that for a long time. Okay, so we're going to talk about a bunch of algal groups, and since you have this PowerPoint, I won't spend a huge amount of time on all of these various details, but these are all groups that you might find in freshwater habitats, and we will probably not see this. The Rhodophyceae, so I think all of these groups that we've got are ending in Phyceae. So phyco means algae, so that's where that comes from. Rhodo means red. And they look red, or maybe a little brown, or maybe a little green, but here's some nice looking, more reddish stuff. But these are all red algae that you might see in freshwater. We usually don't see it around here because they really don't like sediment in the water. We have a lot of sediment in the water. So about 3% of freshwater species, they're often benthic, quote, macroalgae. So this top picture, you can see like chunks of stuff, and if you picked it up, you couldn't see, like, this is what it looks like under the microscope. You could see, like, it would look like little beads kind of on a string that branches, so you can kind of even see what you have just out in the field. That would be macroalgae. Most macroalgae could be red algae, or it could be green, or anything else that fits that. Red algae mostly found in moderately flowing streams with kind of lower nutrients, so not here. They don't really like organic pollution, so basically there's animals pooping in the area and that gets in the stream. They're not really into that. Like a lot of animals, like a bunch of humans or a bunch of cows or Canada geese even. They also don't like sediment. And I think I've collected it one time here. Dr. Blanchard also brought me some one summer from Middle Tennessee. But, so if you see anything kind of reddish looking, it can be exciting. But in some streams where they are found as macroalgae, they can be like ecologically interesting in those streams. Right. Chrysophyceae, or the golden algae. They're related to diatoms, and they have in common glass in their cell wall. And but unlike diatoms, their glassy cell wall is like overlapping scales. So a lot of these cells look like little pine cones. Like this is cyanura, which we get in Pacer Pond in the plankton when it's like late fall, winter, and spring. The SEM shows a little kind of how they look like little pine cones. Most often found in, so they might be dominant in places that have kind of a low or moderate productivity. Generally soft water. You know, have we talked about soft water versus hard water in here? You've heard that term, right? Like having hard water or having soft water. Hard water has more calcium or magnesium ions in it. So you might hear people with hard water complain about scale on their like coffee makers or humidifiers or faucets, and that's just the calcium depositing. So soft water does not have a lot of calcium or magnesium. Low conductivity, so conductivity is the ability of electrical currents to pass through water. And what do you know about water and electricity?
It's supposed to be a good conductor.
So that's what we hear, right?
But Dr. B told us that our water specific in around here is very low.
Yeah, so like I always heard that water likes to go through, or electricity likes to go through water. If you have absolutely pure water with nothing dissolved in it, electricity does not move through it. It's got to travel like across all the stuff that's dissolved in the water. So the less stuff that we have dissolved in it, the less, like if you're out electroshocking, you need to jack up the juice a little bit to
You
do
gotta
it.
do that a lot.
Yeah. That's a conductivity thing. So that has to do with, it could be like salinity, it could be any kind of ion dissolved in the water. It doesn't really matter what it is. Low alkalinity. Alkalinity is the ability of water to resist changes in pH. So like if something has high alkalinity, you would have to add a lot of acid to it to get it to change pH, because there's a lot of like buffering capacity. So if there's a lot of calcium dissolved in something, that provides buffering capacity, so it would take a lot more acid to make it change the pH. If it's low alkalinity, it's very easy for the pH to change. So like places that have low alkalinity lakes, if there's acid rain in that area, those lakes would be really susceptible to pH change from acid rain. Anyway, so if you get a whole lot of chrysophytes in an area, it might have some of these characteristics. pH usually a little bit acidic, 6 to 7. If all of these conditions, like so these above conditions, they tend to compete other algae out for phosphorus fairly well. You won't see a lot of cyanobacteria in those types of habitats, for example. They like cold water. And they can create a, quote, fishy odor in water supplies. Did you know fish don't smell like fish? Fish smell like diatoms. And diatoms and chrysophytes are closely related. And they make this oil that's the smell, and fish eat things that eat things that eat things that eat diatoms, and that goes up the food chain or the food web. And so fish smell diatomy. Fish don't smell fishy. So, but chrysophytes closely related to diatoms, and they also can create, if there's a big, like, a healthy population in plankton and that water is used as a water source, you could like filter out all the cells, but that odor might get left behind. So places that, like probably more up north in like Scandinavia, have more Okay. But we can see, I've seen these two in Pacer Pond in the wintertime. Okay, some fun things about chrysophytes. Many genera are what we call mixotrophic. And so we've learned our whole lives that plants just photosynthesize, but some algae, they only photosynthesize, but some photosynthesize and eat bacteria. And if you're like, well, what about Venus flytrap, Dr. Greenwood? Plants don't actually, like, carnivorous plants are not actually carnivores. They don't eat the insects and get their energy. They trap the insects and break them down and get their nutrients. They don't get energy from them, so they're technically not eating them, even though they're killing them. And kind of sucking the blood a little bit, but anyway, so we don't actually have heterotrophic. Well, we do have parasitic plants, but we don't have carnivorous, like, eating plants. Anyway, I'm babbling. So chrysophytes, there are some genera that will photosynthesize, but they'll also eat bacteria, and they can eat a lot of bacteria. And some genera are obligately both autotrophic and heterotrophic, so they have to photosynthesize and they have to eat bacteria. They have to do both. So that's obligately both. They're obligated to do both. Or they can be facultatively both. You know that word, facultative? It's the opposite of obligate. So facultative means they can photosynthesize or they can eat bacteria, but they don't have to do one or the other. They have to do one. It doesn't matter which one. So I kept saying eat bacteria, but other things they might consume in their heterotrophic form. It could be just particulate carbon, like a starch grain as an example. So like not a whole organism, but some kind of waste or just a piece of an organism that got released into the water. But they could also eat small cells like bacteria or yeast cells or small algae. And these mixotrophs, they can be primary consumers of prokaryotes and mostly oligotrophic I used an unfortunate choice of words here. Can be the primary consumers of prokaryotes. So what I'm trying to say is they can be the thing that eats more bacteria than anything else in a lake where they're commonly found. And you don't have to remember these rates here, but as an example, Denobrion, this thing up here, we get this in Pacer Pond. Any one cell could eat 3 bacteria every 5 minutes, which is faster than a lot of other bacteriivores. And there's another genus, Ochromonas, which is this one, and it's been clocked at 180 cells per hour, so 3 per minute instead of 5 minutes. So some pretty scary bacteria murderers out there. Telling you.
Have you found them while working?
Like while they're eating bacteria?
Or have you found them like when looking around, or are they hard to find? Like with the red
This
ones?
is really common in the winter in Pacer Pond. I've never seen Ochromonas before, but Denobrion's really common in cooler water. Like, it's at Real Fit too. Later in the year. It's really fun to look at. All right, our next group, the dinoflagellates or the dinophyceae. And they are the whirling flagellates. So in the word dinosaur, dino means terrible, dinosaur, terrible lizard. Dinoflagellate, the dino means whirling also. So when they swim, they go in a spiral. And I saw this show about language, and they said if you ask someone to define a spiral, they can't do it without doing that. They have to, like, they can't describe a spiral without doing that, which I thought was interesting. Anyway, so dinoflagellates, when you look at them in the microscope, you might see they have these, some of them have these armored plates, which is kind of funky. Even without an SEM, you can kind of get a sense of those armored plates sometimes.
Is this the guy we saw that's on the whole time?
Yeah, we saw one that was like that shaped, I think. They're mostly lentic, so found. What's lentic versus lodic?
Still one.
Yeah, so lakes and ponds. They can sometimes form blooms, but they're not toxic in freshwater. But red tide, which is in the ocean, that is caused by dinoflagellates. And they tend to max out in the summertime. They're also mixotrophic. And some dinoflagellates they don't even have pigments, they don't photosynthesize. They just eat other stuff. Alright. Good with dinoflagellates. The Euglenophyceae, I think we saw Euglena on Slime TV. So this group, they're all flagellated, and I think it's about 30% of the species are photosynthetic, and the rest are just heterotrophs and do not have pigment. And they tend to be, if they're really abundant, they're probably indicating some organic enrichment. So like a farm field, like a ditch that drains a farm field, you could expect a lot of Euglena in that. We see quite a bit in Pacer Pond, we see quite a bit in Reelfoot Lake. And they're recognizable if you're not colorblind because they have a red eye spot. So their pigments are very green, like grass green. They actually got their chloroplast by engulfing and enslaving a green alga in their history. So they're a grass green color, and then they'll have a red eye spot somewhere. And the eye spot is thought to be able to like detect light, not like see images, but you know, I photosynthesize, I'm hungry, where is light? My eye spot says this way, so I go that way. There is a species that makes a lot of red pigment called Euglena sanguinea, sanguine for blood, like exsanguinate is to bleed to death. And if that blooms, it can look really alarming. You get a bunch of really red water, and people might think that something really bad happened, but it might just be this Euglena, which is totally harmless. Our next group, so this is kind of a different group because these are, this is another macro alga. When you see them, they look like little plants. And so the Charophyceae, and they're very closely related to land plants and look like little small plants. And I've not seen any around here. This stuff is also known as skunk wort because if you pick it up and kind of crush it, it smells skunky. And so these look like little plants, but these are actually just really fat filaments. So they're just kind of weird. We often find them on beds of sand or silt, either in lakes or streams. They have to be somewhat stable beds or they can't like establish themselves very well. They like hard water. And they're pretty good at living in deep water. They're good at getting light in dark places. And they can get all encrusted with calcium, and so we say they can precipitate calcium. And when that happens, it's basically like these little stony little plants, and it forms these big like calcium beds, and that's referred to as marl. And we don't have those around here as far as I can tell. Alright, we got one more weird group and then we'll get into the greens and the diatoms. So, last group, the Cryptophyceae, and we did see that in Pacer Pond Plankton, Cryptomonas. It's the one that had the weird, I called it the little shoulder, and the flagella coming out there. So that weird shape was pretty distinctive. And so there's the Cryptophyceae, also known as the Cryptomonads. Monad just means a flagellated cell. And they might dominate plankton in lakes that are frozen over. So if there's ice cover, they may do pretty well, and just in the late fall or early spring when it's still pretty cold, they can do quite well. And they also like kind of deeper water, which if they can handle dark and cold, then deep water will also work for them. And they are a good food source. Alright, so all those groups, the euglenids, charophytes, cryptophytes, dinoflagellates, chrysophytes, red algae, they're around, but they're usually not going to dominate around here, but we might run across them. So I wanted to mention them. We spent a lot of time talking about cyanobacteria because we always see that. And then our next two groups, the green algae and the diatoms, those are like the other two really big groups. So if you remembered only a few things about algae, just remembering that cyanobacteria, green algae, and diatoms are the big groups that we see. That would be good. Alright, so green algae, or the Chlorophyceae, closely related to land plants. So land plants did not evolve from the green algae that exists today, but they share a recent common ancestor. But they do have that like grass green color. Since they're so common, they're very important primary producers. In a lot of aquatic places, they're kind of like making the vegetables or the produce stand of lake and stream bottoms or plankton. And they are a lovely food source. So cyanobacteria, crappy food source, green algae, good food source. And we've got filamentous forms, we've got colonial forms. There's a lot of diversity in the green algae. And so a few more pictures about that. So the frog spit that I mentioned, that metaphyton Spirogyra, which we saw, is a common member of frog spit. But there can be some other stuff in there too. Spirogyra is really easy to recognize because you have that spiral chloroplast in a filament. And there's nothing else that does that. We might get some colonies that are a bunch of flagellated balls that are hanging out together. And those flagellated balls like nutrient-rich water. So if cyanobacteria haven't taken over, but there are a lot of nutrients, you might get a lot of these colonial flagellates. This one's kind of a fun one because half of the colony has big cells and half of the colony has small cells, which is kind of fun. And it's a sphere. We have this group of green algae called desmids, which are special to my heart because they're so pretty. But they tend to be found in more acidic lakes and wetlands. They also don't really like very hard water, and we actually have quite a bit of these around here. We do have some calcium or something in our water because I always get scale at home when we have well water. So we do have some hardness, but it's not extreme enough to keep out this group of algae. So I could talk about desmids all day, but I will stop there. All right, and one more green I think I need to talk about, and that is Cladophora. So this one I wanted to spend some time on. So Spirogyra, the frog spit, that stuff is really slippery. Like if you pick it up, that's the stuff I was sneezing out of my quote nose in the video that Euryotic. But Cladophora is really cottony, and if you squeeze it, it's very dry. And that makes it kind of a good habitat for things to live on. So the slippery Spirogyra stuff, it's difficult for algae to live on that because they'll just slide off. But you can get a lot of algae that lives on Cladophora. And so if you've got like rocks where you've got Cladophora growing, you can get a bunch of algae growing on that, a bunch of diatoms. And then you can get invertebrates that live in all of that Cladophora because it's growing a bunch of food. And so you can get kind of a lot going on. And if you've got a rock that's covered with Cladophora, it can increase the surface area of that rock by like 2,000 times because of all those little hairs that are coming off of that basically the Cladophora. And Cladophora can bloom. There's a big river in Australia, the Murray-Darling River, and it goes for like a couple thousand kilometers through Australia. And they had a bloom of Cladophora. They might still have them every year, I don't know. But I remember one year, someone that was giving a talk at Society for Freshwater Science meeting, basically they had a Cladophora bloom in that river that was like 1,000 kilometers long. And so I was pretty happy. Cladophora likes a lot of phosphorus. So this is just something that you might see it and squeeze it if it's cottony. You've got Cladophora. Alright. That's all I'll say about the green algae. On to the diatoms, or the Basilaria and these are lovely to look at. They have glass cell walls like their cousins, the chrysophytes, but they don't have the scales the chrysophytes have. But they have a lot of ornamentation to them. So this one, the cell is dead. All the cell contents have been cleaned out, so you can see the structure of the cell wall. But you see all these like lines that we call striae, and this big line in the middle called a raphe, and like the shape and the pattern of all that stuff is what we use to identify species. So it's fun to look at them. They're very lovely. And so diatoms, they need a lot of silica, so that has to be a nutrient that's available to them, or you won't get a lot of diatoms. And they are also a very important primary producer in a lot of aquatic systems. So I mean, we saw a lot of that green algae in Cane Creek, but there were some of those shady spots that all that brown fluff. I mean, that was all diatoms. Like, so that's a lot. That's a lot. So that's important. And they're also a very good food resource. So green algae, they're like plants, they store starch. If they photosynthesize more than they need, they'll chain those sugars together and make starch. If the diatoms have extra photosynthate, they'll make oil. And so that makes them like the pork chops and steaks of the aquatic world. And so they have a lot of calories in them, a lot of good nutrients. So a lot of stuff likes to eat diatoms. So diatoms and green algae, good food. Cyanobacteria, bad food. What else? So we can find diatoms in all different kinds of water, like habitats, all those microhabitats we mentioned, different types of water chemistry, different pH levels, different nutrient levels, but you just might find different species with different water chemistries. And they're usually the dominant group in undisturbed systems. And one of our big ways we disturb systems is adding nutrients to it, which brings in the greens and then the cyanobacteria. But if it's a relatively pristine area, you'll get mostly diatoms living in that system. And we use diatoms in biomonitoring, which I think we'll talk about later this semester. Basically, using what we know about the preferences of all these different algal species and collecting a sample from a stream, look at the diatoms we have and use that information to tell us if the stream is healthy or not. And if it's not, maybe what the problem might be, like nutrients or low pH or sediments, something like that. All right. And one more slide here. So now that we know a little bit about diatoms and green algae and cyanobacteria, there's kind of a trend that we can see in phytoplankton. And this happens over time, and it could be like within a year in a season, or it could be over thousands of years as a lake ages. So lakes are formed and they exist and then they get filled in over time, unless the bottom is dropping, of course. And so keeping in mind that diatoms need silica and nitrogen and phosphorus. Green algae do not need silica. They do need nitrogen and phosphorus. And cyanobacteria just need phosphorus because a lot of them can fix their own nitrogen, right? Okay. So think about, so just kind of over time in general, sort of the first, and this could be starting in spring if it were just over a year, or just like the lifetime of a lake in the beginning. You might start out with diatoms, so phase one you get diatoms, and then diatoms would increase over time. And you can also think of this as what would happen as phosphorus increases. So as time goes by, or as phosphorus increases, diatoms would be the first things to increase, most likely, because they were probably dominant to start with. And then, as they're increasing, they may eventually use up all the silica that's available in the water column. If we were looking at a lake that had turnover, like spring and summer, or spring and fall turnover, turnover can replenish silica back into the water. So silica might be hanging out in the sediment, and in the summer diatoms might use it all up, but then over the winter the turnover would bring it back and they could start again next spring. Or it could just be over time in general, silica gets depleted. Diatoms, when they live and die, they take a really long time to break down, so silica can get kind of tied up a little bit. So anyway, when silica crashes, then your diatoms are also going to go down. And then the next algal group to come in would be the green algae. They tend to, if they have enough nitrogen, they tend to do better than cyanobacteria. So they would be the next ones to show up. And as they increased, what's going to happen to nitrogen?
It goes down.
That's going to go down. So nitrogen goes down, and now which algal group is going to be happy? Cyanobacteria. So silica's gone, nitrogen's gone, cyanobacteria doesn't care, just give me the phosphorus and I'll go crazy. So that kind of like succession of algal communities is pretty common in lakes, like from spring through summer. We often see that kind of change. So the plankton in Pacer Pond, you'd start out seeing more diatoms and other things, and you might get a bunch of green algae, and then cyanobacteria would come in. So that's kind of typical. Or as a lake is aging and getting more nutrient rich, you can also see the same trend. Groovy, groovy. Let me do that slide I skipped at the end of the cyanobacteria. And we'll probably stop there, and then in the lab we'll talk about fungi and so this was connected to, just to remind you where we were when I was going to talk about this. We were talking about that some cyanobacteria have special cells called heterocytes that fix nitrogen. There's some other fun cells that might have, these big fat suckers right here. Those are also special cells that are called achenetes. There's some more right there. And those are resting cells, and they make these when they're stressed. And then those achenetes might like fall to the bottom of the lake, and they could just hang out until conditions get better, and they'll come back. So this would be the way that they survive the winter when things are more favorable for other types of algae. And there was, this is a really neat story, and not something that you might expect. And so there was this lake in, I think it was New York, that was not in an area where nutrient enrichment was a problem. So it was in a like pristine watershed. Everything was great, but they were seeing in the lake itself that algal productivity was increasing. And so it looked like there was somehow nutrient enrichment going on, but there was no source of it from outside the lake, and they couldn't figure it out. And so Dr. Carey, I think Caitlin is her first name, I think, and she's a professor now at Virginia Tech, but for her graduate research, she worked on these lakes and found that the plankton had a lot of this stuff in it, which is called Euryotic. And this is basically, probably don't remember koosh balls, which were basically like a bunch of short pieces of rubber band made into a little pom-pom, and we throw them around at each other in the 80s. Anyway, so it's basically a bunch of these filamentous cyanobacteria, and in the middle they've got heterocytes, so they can fix nitrogen, but they generally just kind of float in the plankton, and this stuff was blooming, and they couldn't figure out why it was blooming if there wasn't supposed to be a lot of nutrients in there. And what they figured out, and like I don't know why the gliotrichia, like the gliotrichia would have had to start becoming more common for some reason. I'm not sure why that happened, but they figured out that when so this would do its thing in the summer, and then when it got to winter, it would make those achenetes, and the achenetes would go to the bottom of the lake, and those achenetes would suck up a bunch of nutrients that were just kind of stuck out or hanging out in the bottom in the sediment. And then in the spring, when turnover happened, that would bring those aconites up into the water, and they were bringing a bunch of extra phosphorus with them, and then they could use that phosphorus. Remember, they can store, extra amounts of phosphorus. They would use that and basically like introduce that into the biogeochemical cycle in the plankton. So these algal colonies were ultimately like scooping up phosphorus from the bottom and putting it into the water column, where before it had just been like stored in the sediment down there. So you're seeing this like eutrophication of a lake from just algae being weird. And so that actually increased the amount of nutrients in the water when it wasn't even human activity.
For once.
I will stop there, and then the macrophytes and fungi lecture is like 11 slides, and a lot of them are pictures, so it